Well, I am pleased to be hosting Dr. Khai Ang to discuss his recent PLOS ONE Lab Protocol paper entitled Staining and Resin Embedding of Whole Daphnia magna samples for micro CT imaging enabling 3D visualization of cells, tissues, and organs, and the associated detailed protocol in protocols.io. Dr. Khai Ang is an assistant professor in the Department of Pathology at Penn State College of Medicine. After earning his Bachelor in Science degree in 2003, he began graduate school researching the molecular phylogenetics of the orang asli in Malaysia, which was the first phylogenetics study among the Malaysian native populations. In 2009, he earned his PhD in genetics from University Kebangsaan in Malaysia. As he pursued his population genetics research among the indigenous people across the world, he realized access to uncontaminated water was a challenge in many of these places. Therefore, he now hopes to delineate the problem of environmental toxicity assessment through the study of morphological and cellular changes in aquatic microorganisms using soft tissue microcomputed tomography imaging to amount to 3D histology. His current project focuses on creating a histological and 3D reference atlas for the water flea Daphnia magna, which has been used in toxicological studies. These developmental reference atlases will serve as a foundation for environmental monitoring and toxicity testing of long-term exposures to different environmental pollutants, even at low concentrations. Khai, thank you so much for having this conversation with me today.
Thanks for having me here.
So, to kick off, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Sure. So in my lab, we are interested to use Daphnia as a model organism for environmental toxicology. So for this protocol, we are trying to stain our Daphnia so that we can use X-rays to image it. One of the key things that we are trying to do is that to use X-rays so that we can look at a whole organism and in 3D rather than a histology, which is a more commonly used method. So for this, we want to achieve a few things. First of all is to achieve consistent staining across Daphnia of different ages. So here we try to optimize a protocol that we can stain this Daphnia, and then after that put it in front of X-rays and scan them to get a 3D cellular or histology images of Daphnia so that we can have an unbiased phenotyping of this organism. And we have to optimize it for Daphnia that are in juvenile and also Daphnia that has are pregnant with or the embryos in the brood chamber, as you may guess that when it is bigger, then it takes a higher concentrations of the staining material here that we use PTA or in Daphnia. So, and also to increase the durations of it.
And so for us to understand better the significance of the protocol and the motivations to optimize it, can you elaborate on what was the state of the art when you decided to embark on this project? What were other methods that already existed but didn't work for you, so you have to develop this new methodology?
Yeah, there are various methods that's been published out there, or earlier on, as I mentioned, one commonly used method is to, when we want to assess pathology in any model organism, is to use histology. H&E staining is commonly available to people. However, H&E stainings require you to sections into these model organisms that you use is destructive. You cannot go back and look at this again as the technology progress. Our micro CT is a method where we can image the whole Daphnia without sectioning into a model organism without destroying it. And if the camera, for example, the camera technology improve or the lens technology improve, higher flux of X-rays are available, we can take the same Daphnia and re-image them. Now, there are protocols that are available for different model organisms too, for a micro CT. Zebrafish protocol has been published. But to be able to use micro CT for Daphnia is a little bit tricky. This Daphnia do have a carapace, and penetrations of PTA varies from organisms to organisms. And this is the first time that we will optimize the protocols in a crustacean model, or especially Daphnia magna with a carapace, and we need to optimize that. And one other things that we have to optimize is that since Daphnia as carapace, they live in water, we want to be able to preserve the structures too as we euthanize this Daphnia. We want to make sure that the carapace doesn't balloon up, it doesn't open up, and it doesn't preserve the original structures anymore that might create a bias here. So we have to optimize all these protocols at every single point, and that's what drivers to create this and to make it available for people. We want consistency in staining and also in fixations of this protocol, especially for Daphnia that right now it's emerging to be one of the model organisms for environmental toxicology.
And going a little bit deeper into the protocol itself, as you were developing the protocol, what were the highs and lows? Like, for example, is there anything that you tried during optimization that didn't work and didn't make it to the final version of the protocol?
Yeah, so we did a lot of things. Or one of the things, it's also when we started, we just fix it in formalin. It's a very common fixative that people use for all model organisms. But when we use formalin or for Daphnia, the carapace just start to balloon very quickly. It start to open up. Or there are different things we thought that Daphnia is a small model organism, is a couple millimeter in size. We use low concentration of staining material, or our staining here PTA, and it works for the small Daphnia when we started that, and then we start imaging our small Daphnias, and it was great. But when we start using the same concentration for a pregnant Daphnia, all our neonates or the embryos in this brood chamber wasn't stained properly. There are spots that we cannot see. And here we are already prepare all these samples, and we were at a imaging trip. We did not get good imaging data. As you can imagine, that becomes a failure in that trip. It costs a lot to go to a synchrotron. It costs people's time. So we want to prevent that in the future, not just for us, it's for other people who are going to use this Daphnia to be aware of that. And we have to come back and start optimizing that. And to be able to get to the right concentration and also at the right size, at the same time, not to increase the durations for too long, a protocol duration. So we have to balance between all this. So when we realize we will have to do all this and achieve that consistency in staining, that was a really good feeling to be able to come to that and to ensure that every time we go for any imaging trip, we are achieving the right staining concentrations for every single animals, and we can see all the different cell types that we want to see.
So, in addition to fixation and staining, what are other critical steps in your protocol? Like any parts that are particularly tricky and others might make a mistake on.
Yeah, so few things that we want to be careful of is that we want to make sure that our chemicals are fresh, or every time we go through different steps of rinsing, or we want to be consistent with that. Those solutions are not too old, or and even in the staining procedure or this PTA that have been made, we want to make sure that there are no sediments. If there are sediments in our solutions and when you stain it, that might be a challenge too, or we might get spots when we image it in using X-rays. And the other critical thing is that we transferring of samples, and we did put a note there in our protocols too, saying that be careful when we handle the samples. We don't want to pipette it too fast, too quickly, or you will destroy the integrity of the samples. Be aware of, for example, the swimming antennae of this Daphnia. If you pipette it too hard, then you break them. Or and we want to be able to carefully move from one tube to another tube. The other thing that we do here too is that we encourage people to use a flat bottom tube rather than like a conical tube, like a Pandolf tube, so that the exchange of solutions into this model organism, the Daphnias, are be able to be better. When it's conical at the bottom, the Daphnia always it will be stuck at the bottom, and when you swirl it, it doesn't move that much. So a flat bottoms will move better and doesn't basically hold the Daphnia down towards the bottom of the tube. So these are a few critical things, and we put a note there to make sure that this is important to watch out, and those timings are also critical. There are reasons why we said it is 48 hours here, and this is 72 hours. And we have tests between 48 to 52 hours, let's say 60 and 70 and 70 hours and we try to be consistent to tell here this is the number of hours instead of writing that 2 days or 3 days, which sometimes can be vague. So we have been really trying to be careful here. This is important, and we are saying hey, this is 48 hours and this is 72 hours.
So your protocol is relatively young. The paper was published just at the end of last year. So it's been
That's
out
right.
for a couple of months. However, I want to know, since the publication of the protocol and the paper, have you made any optimizations, further optimizations or changes to the protocol, or you currently use it as it was published?
We have not made any changes since then. We have used it as it is since published. We have optimized this protocol for the last couple years actually, to make sure that this is consistent. We get it out. We have not done quite a lot. Or one thing that we have done over the last couple of years too is to think of materials that we normally in a wet lab, what we already have, or to make sure that when we get this out, it is optimized for people too. Or if you were to go into the protocol, you will see that we have even published a part where how to hold these samples for imaging using a pipette tip instead of a Kapton tubing, because that is something that's available in most of the wet labs. So in a way, we have been trying to optimize this the best we can, and when we publish this, and since then we have not received any comments on how to need for us to optimize it yet, or challenges that when people use this protocol, or there are things that maybe it's not optimized for them. But if people do find if they have any questions or things that they improve on, feel free to reach out to us, and then we are very happy to talk to people on how we can optimize this together. At the end of the day, it is for the community too. If we need to update this, and we will.
So my last question regarding specifically protocol is, I mean, you just mentioned that you have been using this protocol for about 2 years. So have you used this protocol to collect data related to environmental pollutants and toxicity already?
Yes. So we have worked with different things. Recently, we just published a Daphnia atlas. It's histology. We are working on a 3D atlas right now. We have actually used the same protocol. We expose Daphnia to environmental toxicants, or for example, glyphosate. Or we have exposed our Daphnia to acetaminophen and image that using micro CT2. And we are working towards publications of these results and also a 3D atlas. So we are constantly using this or trying to identify different environmental pollutants or toxicants that are in our environment and expose using Daphnia as one of the sentinel organism, expose them and image it. Or we have been able to look at very fine details or cellular resolutions for this Daphnia. I think that was a paper we published just a couple years ago that compares our Daphnia microCT to acetaminophen, or just a figure in a paper, and that's also available.
And I would like us to change gears a little bit now. And I want to know about your motivations to send the protocol to PLOS ONE to be peer reviewed as a standalone methodological paper.
Yeah, we it is always one of the goals in our lab is to make our science publicly available, and PLOS are one of the great thing about it. It's available to people. It is free for access. And why we choose PLOS and make it that they can get it out to everybody. Why we want a peer review is also because it is not just for us in our lab, and we want to make sure that the rigor in science and the reproducibility is there too. There are a lot of experts out there. There are people who use micro CT for various research. People have optimized protocol for chicks, for zebrafish, for xanthopus and different organisms. So it is one of the ways for us to put these things together, get it peer reviewed to make sure that what we are doing it can be reproduced by our peers or make it available for other people. What good it is if I were to develop this protocol and just keep it for my lab. I think in it's cliche, but science has been moving towards our international collaborations. The impact of it will be more, or if we make it available to people, and Daphnia has continued to emerge as one of the organism in environmental toxicology. So when we make it available, PLOS ONE, peer review, then the impact is there, or not just in what I'm doing, but overall in the environmental toxicology. This will be a method for people to use. One of the things that keep coming up right now, even in the molecular world, people are doing spatial transcriptomics. But one of the things that we can do as we publish this 3D imaging protocol is to enable people to correlate the spatial transcriptomic data later on and back to a 3D file. If we have Daphnia that people can scan it in 3D and people who do spatial transcriptomic, these are things that later on, or when it comes to analysis, you can link two results together. So we're just trying not to keep it with us, and realize that PLOS is also a good way to go forward, to do this.
And since you mentioned, peer review and validation, how did the feedback that you received from the peer reviewers shape the final version of the protocol?
Yeah, appreciate that questions. So when we get it out, things when we write a protocol, sometimes we get these tunnel visions. We think that we have done this right, but actually peer reviewers have also helped us to make it tighter and polish in our wording, and sometimes asking us specific questions. What do you mean when you say a specific terms? And these are experts in our field, and also challenge us at the time, saying, have you looked into if we look at embryos from age instar from 1 to 3, what about instar 4 to 7? What about this specific time point? Can you look into this further? And we work together in that way. And we have looked into all that, and we just need to be able to tighten all this out and get the paper or make it more understandable for everybody. So that's really helpful. And, and to be able to put in specific, or not terms, but clarifications in the methods that we are doing. Sometimes we forget we have done this so much, and we just say, fix it in, for example, Boolean, and we forget what's the concentration. So peer reviews have been able to help us to really tighten everything up, which is really, really good.
And the final question that I have, although it might still be a little bit speculative because, again, this is a young paper, but what has been, or what do you think will be the significance of having this methodological paper published for your career?
Yeah, my lab is interested to continue to build an atlas, a Daphnia, or a biological atlas for the research community. So we have published the histology protocol, and right now this is the 3D protocol. And one of the things, as I mentioned earlier on, is that there are new technologies has been developed. So this is the start of, or it might be young, but people have been using this for some time to make it accessible to general researchers and all scientists, are where later on more people are going to start interrogating their model organism in 3D. No longer a specific organs, no longer just a specific sections using histology. Here we can look at a whole organism in 3D. So this will be significant when we can make these protocols available for people to do that. And we can look at volumetric changes. We can quantify volumes in 3D. So if a cell swells, or now we can use this protocol, image it in X-rays, and look at it. So in that sense, I think this will be a very important steps, a protocol for people to stain Daphnia properly and image it, or help people to achieve that. This is going to be also a protocol that will be listed in our atlas, make it available for all people. We hope an atlas that we are developing right now to be a central place on our whole organism phenotyping for Daphnia community. So this will be part of it, that we want to collect everything in one place for people to study that. It enables a lot of other things. I mentioned volumetric, or this protocol will enable, with the progress of AI, we can do organ segmentation, for example, of different organs. I think we show in a protocol here that we can section out the ommatidia, the eyes of this Daphnia, and we can do a lot of other organs in here too. You can only do it using a 3D protocol, not very hard to do it on a 2D, because you want to get the whole volume of it. So there's going to be a lot of this, and this is just a first step. We are optimizing for Daphnia. We are doing it for other model organisms, and we are collaborating with other investigators too, who are interested to look at the same file or same exposure and then do spatial transcriptomic. Right now, we're going to interpret the data not just only on a single cells, but where is these cells located in 3D? What is the adjacent cell type? What is the adjacent organs and tissues? So we, with a 3D data, we can interpret our data in a spatial context.
Khai, thank you so much. I don't have any other questions. I don't know if there's anything that you would like to add that we haven't touched so far.
So thank you for chatting, and I hope that people who use this protocol, if they have any questions or need any help at all, feel free to reach out to us. We would like to work with everybody. If we need to optimize these protocols, as you asked earlier on, if there's anything that we have to make it better for people, we hope to do it too, and then we can work together to make it better for the community.
Thank you.
Thank you.